All right. Hello, everybody. If you watched the first half of this video, welcome back. But if this is the first video that you're watching, hello, my name is Max Hammer. I'm an MD PhD student at UCSF, and I am here to talk to you guys today about mutant crops and how we genetically engineer plants to make really cool different mutant organisms that we can use for food and for medicine and for other really cool things. Um, so yeah, hopefully everybody can now see my lecture slides. Um, and we'll start this talk on mutant crops. So I'm just going to start with a really brief overview of what DNA is and how it's used as instructions to make up cells and pretty much everything in our bodies and plants and every other living thing. So DNA is the basic way that cells and living organisms store information. So DNA here, we see a cell, and within that cell, we've got these things called chromosomes. And those chromosomes are like tightly packed spools of DNA. And so DNA, when you unspool it and look at it at the most basic level, it's just made up of these nucleotides that are these little molecules that come in four flavors: A, T, G, and C. And pretty much the way that you line up all of those letters is just like we do when we're making words, like in English, where you can line up those letters in a whole bunch of different ways to spell a bunch of different words and sentences and store information. So this is what's called the central dogma for DNA, where we have this DNA that has all these letters, and those letters are actually then used by things called ribosomes to build proteins. And proteins are what are kind of like the workers of the cell that go on to do pretty much everything in our cells and our organs and in our bodies. Um, so the way that all of these A's, T's, G's, and C's are arranged will determine which amino acids are linked together to make different proteins. So amino acids are like the basic building blocks of a protein. We string them all together. All of them have different unique properties, and the way that they're all lined up makes different proteins that have different functions, different properties. So that's how DNA works. And what's amazing about DNA is that every living organism has DNA, and every living organism uses DNA in the same way. So think of this like if you've got a whole bunch of books in a library or a whole bunch of instructions for how to, say, cook something in a cookbook. All of us can share those books with each other, and all of us can read those books because we all use the same language. I guess if we're all, say, in an English-speaking library, and we can do the same with different organisms. So what's really cool is that we can then mix and match these genes. So if we have a gene which is just one unit of instruction to make one protein within a cell. We can take genes from one organism, say taking instructions from, say, I know how to cook cake and I want to tell you how to bake a cake. I can go and give you my book and then you can bake a cake. So we can take those genes from one organism and then have another organism read those genes and make those proteins. So here's a couple of really cool and crazy examples that were from these kind of first experiments of mixing and matching genes. So this first one over here of these pigs and these mice is for a gene for a protein called GFP, stands for green fluorescent protein. And GFP is originally produced in a jellyfish that glows down in the deep ocean. But we can take that gene, the DNA, that has the instructions to make that fluorescent green protein, GFP, and we can just put that same gene into the cells of, say, a pig, and then the pig cells will be able to read that DNA in exactly the same way, make that gene in exactly the same way, following all those instructions, and then the pig cells have this glowing green protein. So here are some examples of these pigs and these mice that have that GFP gene put into them, and then they glow, which is so cool. And these are just some other examples of different genes. This is a gene that makes muscles really big, or even the genes that grow an ear. This one's a little bit more complicated. There's a couple more genes involved in that one. But the same idea that we can take genes from one animal or one organism or one plant, put them into a different cell, and that cell can read all those instructions and make the protein in exactly the same way. So those were all examples in animals, but plants have DNA and use DNA in all the same ways that animals do. So here is just a really, really beautiful picture of some plant cells, and you can see these chromosomes inside of the plant cells. So all of these chromosomes, these like wiggly fingery looking things, those are big spools of DNA inside the cells that you can kind of unspool just like in that first picture and read them out like the instructions for how to make proteins. And just like in humans and in other animals, that DNA gets read by these cells, they make proteins, and then they use that to build up the whole plant. So, just like how I showed you with that green fluorescent protein gene, where we put it into pigs and into mice, we can do the same thing with plants, where we can take a gene from one plant that we want in another plant and just insert that gene into the other plant. But that is all nice and fun to talk about, but how do we actually do it? So there's a couple of different ways that scientists have figured out to get a gene from one plant into another plant. This first way, I still find really crazy and really wild that we do this, but we can use things called gene guns, which sounds super sci-fi and futuristic, but it's real. I'm here, we've got a picture of one. It's literally like a gun that you hold with your hand. And what we can do is take these tiny little beads, these DNA coated gold particles, as they're called here, so little beads of gold that we wrap with DNA of the gene that we want to insert into the plant, and we just shoot the plant with it. And what's amazing is that that DNA flies into the plant, crashes into the chromosomes, and then just gets bam inserted into that DNA, inserted into the chromosome. And then the plant has that gene, it can read those instructions, and it can make whatever protein we want it to make. Now, there's some slightly more sophisticated ways of getting genes, and you can imagine the gene gun's a little bit messy. So there's other ways to take DNA and then use different methods to get it inserted into the cell. So one form is called transfection, which is where we take the gene that we want, that DNA, and we actually put it inside of a virus, because viruses have evolved for millions and millions of years to be really good at taking their DNA and putting it inside of cells. So we can take advantage of that and take a gene that we want, put it inside of a virus, and have the virus do all that work for us of inserting that gene into the DNA of the cell that we want it to. Here's a little example. We've got these little rings. They're called plasmids that are instructions to make a gene or instructions for a gene to make a protein. And we just put that inside of a virus. In this case, it's the gene to make that green fluorescent protein. Then we have that virus go and infect a cell, and the virus uses all of its machinery that it's evolved over millions of years to insert that DNA into that cell, and then the cell makes the protein. There's another method for this where for certain types of cells, like bacteria, we don't need the virus. We can just put what's called a plasmid, so that little ring of DNA, into some water with the virus and either shock it with heat or with electricity, and the virus kind of opens up for a second and sucks in the DNA. But that only works for certain types of cells, but it's another approach that we can use. In a more modern invention, you guys have probably heard about CRISPR. If you haven't, no worries. But so CRISPR-Cas9 is this really cool little mini machine that we've discovered. It's a little protein and machine that we can send into a cell that will go and find a specific region of DNA that we want it to find, and then either cut it out or put a new piece of DNA in. So it's kind of like this little mini machine that we can engineer and have it go and do our little DNA bidding inside of the cell. So here's just kind of a potentially overly complicated picture, just showing different ways that this CRISPR-Cas9 system, we can give it a little guide to know where to find in the inside of that chromosome, and it can do a whole bunch of things like turning genes on, turning them off, cutting them up, sticking a new gene in. It's really amazing, and it's really revolutionized the way that we're able to tinker with and engineer DNA inside of cells. So we're going to now talk about using these tools to change crops and plants in ways that we want that are beneficial for us as humans. But I want to start by kind of taking a little short history lesson about the history of genetically manipulating plants. And we've been doing this for a really long time. So old school genetic manipulation of crops, for hundreds of thousands of years, humans have been growing plants, eating them for food. And what we've been doing is slowly over time, we grow a plant, and we grow a whole harvest of them. And let's say that we're trying to, grow a tasty fruit on that plant. And we need to pick some seeds to plant for next season. And what humans would do is they would say, of my 100 apple trees, I'm going to pick seeds from the tree that had the biggest apples. And I'm going to use those to plant for my next round of trees. And over time and over time and over time, we're kind of gradually selecting for bigger and bigger apples. And what's going on is that what causes those changes in apple size is actually changes in DNA, changes in the instructions for how to make an apple. And after many hundreds and thousands of generations, we've just been slowly letting evolution and nature make those changes for us at random. And we've been just pushing them along in the direction that we want. So this is a really cool example of a handful of vegetables. You guys will probably be familiar with at least a couple of them. Cabbage, Brussels sprouts, kohlrabi, kale, broccoli, cauliflower. All of these vegetables, which look pretty different, all actually came from the same original plant that humans used to grow and eat many hundreds of thousands of years ago. And based on what they were kind of trying to pick for their next generation of crops, they exaggerated these different parts of the plant. So, for example, selecting for these terminal buds made these really big cabbage plants, or like selecting for a really big root if you wanted to eat the root, made this kohlrabi, like this big old root. Broccoli is actually flowers. The part that you eat of the broccoli is flowers. So slowly selecting for those flowers eventually turned this plant into broccoli. But so this is a great example of how humans have been able to slowly select for different features that they want. And through random mutations of genes, it's able to slowly shape the instructions for that plant. So we've been genetically modifying plant species for as long as humans have been growing plants. We just didn't realize that that's what we were doing. But it was always genes underneath in those cells all along. It was always DNA. So I just want to go through and show a couple of, in my opinion, really interesting glow-ups for different fruits and plants that we used to eat. So here's an example of watermelon, these big, fat, juicy melons that we eat these days. But they actually started off being really small. Most fruits started off being really small because they don't need to be that big in order to survive and plant their seeds and have animals come and eat them for the plants to survive. So down here at the very bottom, these tiny little melons are what watermelons originally used to be like before humans started growing them. And over time, we've slowly selected them to be bigger and bigger and have more and more fruit within them. Until now we have these giant, juicy watermelons today. Similar story with bananas. Bananas used to be like these tiny little things down here at the bottom, these huge seeds, not a lot of fruit. Can you imagine taking like a big bite of one of these bananas today? It'd be terrible. But we've slowly over time selected for bananas that are bigger and bigger, that have more fruit on them and less seeds. And we'll talk about bananas later because I've got some other really cool genetics going on. Here's an example of pineapples. And pineapples we've selected for over a relatively small amount of time, just in the past kind of 400 years. But they used to be these small little nodules. And slowly we selected for those nodules to be bigger and bigger until they were all just one giant fused together fruit, like this pineapple that we see today. Corn's another example where it used to be a grain. It used to be almost kind of like grass, like if you let grass grow too high. And we slowly selected for those kernels to be bigger and bigger and bigger until we have this massive corn that we have today. So genetically modified organisms, or GMOs, is the exact same idea where we've got a trait that we want, say bigger fruit, or bigger flowers or bigger leaves, like in those examples I showed you of like cabbage and broccoli. But we're just using science to do it a lot faster. So instead of having to wait for random mutations in the DNA and evolution to just randomly hit the gene that we want to make the fruit do the thing that we want it to do, we can instead use our knowledge of DNA and genetics to go in, find the gene that we want, and either turn it on or turn it off or put it into that fruit. So one of the first examples of this was done in apples. So everybody's probably seen, if you leave an apple sitting out after you cut it open or after you bite it, it turns all brown and kind of nasty. That is because of enzyme, which is the result of a gene within the apple called polyphenol oxidase. So once you cut the apple, it starts breaking down the apple and doing some stuff with oxygen that turns it brown. So scientists were able to go in and just delete that gene within the apple so that it doesn't have polyphenol oxidase anymore. And now the apple doesn't turn brown. And it worked beautifully. I mean, here's a picture. This was from a picture of the apple with the gene, and then after we removed the gene. And so we're able to see that we can go in, tinker with the gene of interest that we're, want to change, and we can change the fruit. There's a couple of other really cool examples here. There was this disease, this fungus that was killing papayas in Hawaii. And they found that they could change one gene in the papaya. That meant that the fungus couldn't infect the papaya anymore. And so they did that, made this new line of papayas that had that mutation in them, and it was able to save the papayas on Hawaii from all dying from this fungus. Then there's this example of this rice. So instead of turning a gene off, this is an example of inserting a gene and turning it on. So in countries that are currently struggling to have enough food to eat, something that is very easy to grow is rice. And a lot of countries are able to eat a lot of rice, and that's where a lot of their nutrition comes from. But rice doesn't have a lot of vitamins in it. And in some of these countries, people don't get enough beta carotene, which is an essential vitamin. It's important for your eyes to be able to see. And so scientists created this new rice where they inserted the gene that makes beta carotene synthesis into the rice. And now that rice is able to produce a ton of beta carotene. And you can eat that rice and get all that vitamin just from eating the rice. And they can turn it on. The rice has all the instructions to make it, and it makes a bunch of beta carotene. It's really cool. So you guys have probably heard about GMOs before, and they can be a hot topic of debate about whether GMOs are good or GMOs are bad. I've just been telling you all about GMOs and why I think that they're really interesting and really cool scientifically, and why all that GMOs are doing is taking genes and turning them on and off, just like we've been doing for hundreds of thousands of years, and that these genes are not inherently good or bad. These genes are just the things that tell our bodies and our cells how to make proteins like every other gene in our body. But GMOs have still been a really hot topic of debate, and there's a lot of people who are opposed to GMOs, and I think it's important to briefly talk about some of the political discussion about whether GMOs are good or bad. So I want to first start by saying that biologically, GMOs are totally healthy, totally fine. As I was saying before, changing a gene within a cell is just giving it different instructions for how it makes proteins in the same way that all other cells and all other living organisms turn genes into proteins. So the fact that something is a GMO does not automatically make it bad, doesn't really mean anything about it being good or bad. It's just changing a gene. Now, something that is really important to discuss about GMOs is it's important what gene you're changing and how you're going to be using that changed gene. So some examples of GMOs are plants that they have scientists have edited so that you can spray all these nasty chemicals on them, pesticides and herbicides that will kill bugs or kill weeds that aren't, and the genetically modified plants and crops won't die from those nasty chemicals. But that means that then these companies are able to spray a whole lot of these nasty chemicals onto these GMO crops, which then can run off into the environment and do some damage to other insects or other plants. So there's an example of GMOs, which again, biologically, totally fine, but being used for something that can be environmentally harmful. So we need to ask ourselves what the GMOs are being used for, and is that something that we agree with or disagree with? The other big debate right now is about how we legally regulate GMOs. So there is currently a lot of uncertainty about how we should consider ownership of a genetically modified crop. And so Monsanto is an example of a really big company that makes a lot of these genetically modified plants. But you can imagine that plants pollinate with pollen in the air, and there have been cases of genetically modified plant pollen blowing into a different farmer's land and then pollinating their crops, which means that their crops now have the gene. And it's been a big debate as to whether or not then Monsanto can come in and sue that farmer for growing their crops without paying for it. So GMOs can be a very very lively debate as to whether or not they are good or bad, but that is not because genetically modifying a plant inherently makes them harmful or bad. It's all about the ways that we as humans and as a society use them and regulate them. That's important. So if any of you go out and have any debates about GMOs, I want to make sure that you understand kind of the factors at play here, and that it's not that GMOs are going to in some way turn you into a mutant, or those mutant crops are going to hurt you or give you cancer. No, no, no. It's all just proteins, but keep all this in mind. So we're now going to talk about some other really cool ways that we use genetics to alter the foods that we eat in ways that aren't just changing one gene. So we're first going to talk about giant cells, or what's called polyploidy. So I've already mentioned that chromosomes are what contain the DNA that make up all of the proteins within your body. And humans, we have 2 copies of every chromosome, which means we have 2 copies of every gene. But you can wonder what would happen if we had 4 copies or 6 or 8 or 10 copies of every gene. Now, in humans, that unfortunately doesn't work. But in plants, you can do this, and you can imagine if you have twice as many copies of the genes, you're going to make twice as many copies of protein. And so what's amazing is that we can say have a cell that has 2 copies of a gene, and let's say it's this big, and then you double that, and now it's got 4 copies, and now it's going to be this big. It's going to be twice as big. And we do that when we're growing plants that we want to eat, because we want a nice, big, meaty fruit. So an example of this is strawberries. If you guys have ever seen those like really massive strawberries, are like this big, a lot of those are polyploidy, which means they have extra copies of their chromosomes. So here's an example of like strawberries being octoploid. So they have 8 copies, which means they have 4 times as many copies as, say, like humans would have. And what this means is that each cell just makes a lot of extra copies of the protein, and everything just kind of gets scaled up. So you get these huge polyploidy strawberries with really big cells in them. And we do that with other plants as well. We want to eat to make them really big, like those giant grapes, or these giant strawberries, or those like huge blueberries that you'll see at the store. Now, one of my favorite plant genetics things that we do is our ability to make seedless fruit. So when I was a kid, like my whole life, I always wondered, how do you grow a seedless fruit? I'd be like eating this watermelon, and like, I know that you have to plant seeds in the ground to grow a fruit, but this watermelon doesn't have seeds. So where did it come from? How did you plant it? How did it grow? How do you plant the next watermelon plant? And the answer to all of these questions is genetics. So some of you may have learned about mitosis and meiosis in your biology classes. If not, that's okay. But like I was just saying before, every cell has chromosomes, and it has multiple copies of each chromosome. And cells divide, and when you're going to make a sperm or an egg, you want to make half the amount of chromosomes within that cell, so that then when it fuses to fertilize, you then have half and a half that make a whole, and you've got a full copy of your genome. So all of us get 1 copy, kind of half of our DNA from mom, half of our DNA from dad, and then those come together to make one whole person. And then if you ever go and have kids, you're going to give your kid half of your DNA, and then their other parent will give them the other half, and that'll come together to make a whole. So plants do the same thing. But what we're able to take advantage of is the fact that these plants, we can give them all different numbers of chromosomes, right? Like I was just talking about in the polyploidy slide. And there are these checkpoints when you're making these seed cells that are kind of like sperm or egg, where you have to have a nice evenly lined up half of each type of chromosome in order to split and make those seed cells. So what we can do is make a plant where the mommy plant had, let's say, 6 copies of their chromosomes. The daddy plant had, let's say, 4 copies. Then when they come together and that baby plant is trying to make its own seeds, those chromosomes can't line up because there's not the same number. So you have some that aren't matched. And there's these checkpoints when you're making seeds that the cell says, oh no, we don't have a perfect match of our chromosomes, so we can't go on and make a seed. And that's what happens. As we make these crops where we take, say, a watermelon, where the mommy watermelon has 6 copies, the daddy watermelon has 4 copies. We put that together, make a baby watermelon that has 5 copies, and it's able to grow and make a watermelon totally fine, totally normally. But when the baby watermelon goes to try to make seeds, it can't line up its chromosomes properly, and so the seeds just don't work. So you don't grow seeds. And you have to just keep doing that over and over again. So you keep kind of your garden of mommy watermelons that have 6 copies. You keep your garden of daddy watermelons and you grow those every year. And then every year you have to remix those to make your baby watermelons with 5 copies. And we keep doing that, and that's how we make our seedless fruit. Isn't that cool? So here's just some examples of some fruits that we've done that with. So the most common ones that we talk about or that you see are things like grapes and watermelons and actually even bananas. All of these fruits naturally have seeds. Actually, every fruit naturally has seeds. And any seedless fruit is because humans have tinkered with its genetics. And then we just do that crossing so they can't make their seeds. So we're going to talk now a little bit more about bananas because bananas are absolutely bananas and really kind of crazy genetic organisms. So, really neat fact about bananas is that every banana that you see in the US that we buy in stores and that we eat is a genetic clone of all of the other bananas. Every banana that we see in the States is all genetically the same organism. So, what I mean by that is that there was one original banana plant, and we took that plant and we cloned it and we cloned it and cloned it thousands and thousands of times. So it's kind of like if, you took me and you took a little piece of me and you grew another one of me and then kept doing that until there were a bunch of me. And all of us were the same genetically, we're made out of the same DNA, all look exactly the same. They've done that with bananas. So each tree is genetically the same banana. And so every banana has the exact same DNA in it, which I think is absolutely wild. Now, they do that because what I was talking about with the seedless fruit, where the banana can't actually itself reproduce and make other bananas. So what we have to do is do that cloning process where we split it and we grow a little piece of it in a dish in a lab and then we plant it. And that's where we've made all of our bananas. Now, there's kind of a wild history to this where we have done this in the past. Back in the early 1900s, we'd already done this and we had our one single genetic banana that was all over everywhere in the US. And that banana actually started to get infected with this fungus. And because every banana was the same, the fungus spread really fast and killed every banana plant in the US. And that banana plant, those old bananas, actually tasted different and were a slightly different species than the bananas that we have today. So if anybody's ever eaten like a banana Laffy Taffy or like artificial banana flavored candies, they don't really taste that much like bananas, right? Like they're really sweet. They taste different. And what's crazy is that that old banana, that one that is now extinct, tasted a lot more like that banana candy. It was a lot sweeter, and these banana Laffy Taffies were invented back when the old banana still existed. So it used to taste a lot more accurate to bananas, and nowadays it's just that the banana has changed, and so the Laffy Taffy doesn't taste like the new bananas. So we're just going to end our lecture here with a little rapid fire talk of some of my favorite GMOs that I think are really, really cool. So people probably here don't really like going and getting vaccines. It's not really fun. You get stabbed by a needle. Nobody really likes that, especially little kids. But they're actually currently developing fruits that are edible vaccines. So a vaccine is just a little piece of protein that your immune system recognizes, and then your immune system is able to recognize it again in the future when you get sick and infected by that, say, virus or bacteria. So the idea is that those proteins that are used in vaccines are made by genes, just like everything else. And so if we can put that gene inside of a fruit, and then you eat the fruit, and then by eating the fruit, you get all that protein from the genes that the fruit made. It'd be just like if we injected you with a needle and put that protein into you. Instead, the plant makes the protein, you eat the plant, and then you have the protein in you for your immune system to learn how to fight against. So be looking out in the future for our banana vaccines that nobody has to get shots anymore. There's also these really cool pollution fighting plants. So plants are amazing reservoirs for carbon. So you've probably all heard about carbon dioxide in our atmosphere and how it causes this greenhouse effect where it traps in heat on our planet. And that's what's contributing to climate change. And so plants are a great way to capture that carbon out of the atmosphere. They use CO2 to make sugar and then make their plant body out of. So we can use plants to kind of suck that carbon dioxide out of the air and then trap it into plants and back on the ground. So they've made these couple of different species of plants that are really, really good at sucking carbon out of the air and then storing it in really big, fat roots. This is quite possibly my favorite. Well, no, this is my second favorite. My favorite one is still yet to come. But this is a GMO called scorpion cabbage, which is a species of cabbage that originally farmers were really struggling to grow because all of these caterpillars would come and eat it all up. But they didn't want to spray these cabbages with pesticides because, like I said, those can be really bad for the environment. So what they did was they took the gene for scorpion venom that scorpions have, and it has the instructions to make this venom that scorpions use to kill insects. And they put that gene into the cabbages. And what's amazing is that this type of venom does not work on humans. It has no effect on humans. So we can eat this cabbage and be totally fine. We don't even notice that it's in there, but it kills insects that try to eat it. So this cabbage is able to then grow, and the insects can't eat it. It's able to grow nice and healthy. Humans can then eat it, and we don't need any of those nasty chemicals and pesticides. So this is an example, like I was saying, where GMOs can be used for good or they can be used for bad. This is an example of a GMO that's being used for really good things and is actually helping us not need to use those nasty chemicals. So this is a great GMO that's doing really positive stuff. So before you say that any GMO is good or bad, first look at what it's doing and if it's helping out the environment, if it's, this one has no health effects to humans, it's perfectly fine and healthy for us to eat. Like I said, you won't even notice that there's scorpion venom in it. You just get to say it's really cool because you're eating scorpion cabbage, which is a really cool name. And then here's my last one. This is by far my favorite GMO out there. You guys have probably heard of Impossible Burgers or Beyond Burgers, this plant-based meat. And the big problem that like vegetarian and plant-based burgers had was that they didn't really taste like that nice bloody like red meat that like almost tastes kind of like iron, like the taste of blood. So scientists asked themselves, what makes an actual beef burger have that red kind of iron bloody taste to it? And the answer is hemoglobin, which is a protein within our red blood cells. It's what makes blood red. It binds iron. It's what gives it that taste that tastes kind of like metallic a little bit. It's that iconic taste of blood. And they said, okay, great. If that's what makes hamburgers, beef patties taste like meat, let's just take hemoglobin and grow it in a plant. And that's exactly what they did. Is they took this like hemoglobin, which is a protein that is really, really similar to hemoglobin, and it has iron in it, and it tastes really similar, almost exactly the same to blood hemoglobin. And they engineered these soy plants to express it at really high levels, make a ton of this protein in their roots. And what's crazy is you can take these plants and you cut open their roots. You can kind of see it here. And the roots will bleed. Like they look red, like you're cutting open raw meat. Here's another picture of them over here looking really red and bloody. And so then they can grind those up and use them to make these Impossible Burgers with a bunch of other things. And that gives it that nice taste like actual beef. So that's why they've gotten Beyond Burgers to taste so good. And again, here's an example of a really good positive GMO that's helping us transition from eating cows and eating beef over towards eating vegetables, which don't have any of the moral questions and environmental impacts that cows do. So another GMO, these soybeans aren't gonna turn you into a mutant or do anything. They're not hurting anybody. They're just happy little soybeans growing their little bloody roots, and we can use them to make plant-based and environmentally friendly Impossible Burgers. So these are by far my favorite GMO. But that's the end of my lecture. Thank you guys for listening. Hopefully, I've shown you guys some really neat GMOs you can go and tell all of your friends and family about. And I've helped convince you that GMOs are super interesting and taught you a bit about the genetic tools that we use to help manipulate plants and use them to grow really amazing and futuristic foods. But thank you guys for listening. Once again, I'm Max Hammer. I'm an MD PhD student at UCSF in San Francisco. And thank you all for listening. Bye.